Hi, I'm Mike Feinzelberg.
And I am Rotem Perry.
And we're from the Department of Biological Chemistry at the Weizmann Institute of Science. Our group studies long-distance signaling along nerve axons that convey information about injury from the injury site in the nerve to the neuronal cell bodies. This signaling takes place over very long distances. In molecular terms, if we were to increase the size of a neuronal cell body to the size of an average human being, the axon would stretch out over 30 kilometers. As opposed to the rapid electrical signals that convey much of the information in the nervous system, these injury signals are trafficked on molecular motors that move along microscopic tracts inside the cells that are called microtubules.
Several years ago, we identified a key player in the injury response, a protein called important beta-1 that is needed to connect signaling cargoes to motors moving in the direction of the cell body. This finding was surprising because proteins were previously thought to only exist near the cell nucleus, where they shuttle molecules across the nuclear membrane.
Even more surprising was the fact that the important beta was made locally in nerve axons, whereas the prevailing scientific dogma is that axons are not capable of protein synthesis. To find out where the protein was really being produced, we needed a way to focus on important beta-1 specifically in nerve axons.
That is when we discovered that the important messenger RNA molecule in the axon is longer than the one in the cell body. Using very specific gene knockout technique, we removed just that extra segment of the messenger RNA from the mouse important beta-1 gene. When we did this, we saw that important beta-1 in the cell body was unaffected, but there was none to be found in the axons. The response to injury in the mutant mouse was reduced both in the target gene activation and in physical recovery.
In other words, we have shown that important beta-1 is actually produced in nerve axons and that the local production is required for effective responses to nerve injury. We will now be able to use the new mutant mice to study the roles of such local production of important beta and other critical proteins in the nervous system in general.